Good morning, dear students. We continue to tell with you about carbohydrate metabolism, and today we will tell about one more way with glucose, about pentose phosphate pathway, and after that, during the second part of our lecture, we will tell about hormone regulation of carbohydrate metabolism. So let's start. First of all, what is pentose phosphate pathway? This pathway has a few names, also called as gyxose monophosphate or pentose shunt. First of all, this pathway occurs in the cytosol of the cell, and this pathway includes two steps: oxidative phase and non-oxidative phase. During the first phase, we produce pentose, phosphopentose, which gives the name of this pathway, and carbon dioxide, and one more very important product, NADPH2. During the second, non-oxidative phase, happened the interconversion of pentoses into each other, and to conversion of pentoses into gyxoses, fructose 6-phosphate, and trioses, glyceraldehyde 3-phosphate, which are intermediates of glycolysis. Both phases, I again repeated, occur in the cytosol. One more name of, we call pentose phosphate pathway like alternative oxidation of glucose, because during the first phase happened really glucose oxidation. But why alternative oxidation of glucose? Because you know one more way of glucose oxidation, glycolysis, which lead mainly to energy production. This way never lead to energy production. This way we use for the production of another but the same very important substances. First phase, oxidative phase of pentose phosphate pathway. First reaction, you know, it's a general reaction with glucose. We already discussed this reaction many times, and when we tell about glycolysis and when we tell about glycogen synthesis, first reaction, you know, it's a glucose phosphorylation. And two enzymes, dexokinase in all tissues and glucokinase in the liver, catalyze this reaction. We already discussed this reaction very many times. After that, this reaction is not unique for pentose phosphate pathway. This reaction is the first reaction with glucose into the cell. After that, maybe different way for this glucose 6-phosphate using. We can use it for glycogen synthesis, for energy production, or like today, for pentose phosphate pathway. And after a few reaction, we produce two main products, ribose 5-phosphate and NADPH2. NADP, pay attention, is different from NAD with only one phosphate group in one of ribose. In NAD plus we have now here this phosphate group. But these little differences lead to a pair of absolutely new reduced equivalent NADPH2, which have another unique role in our body, which binds with high specific NADP-dependent enzyme. And what is the role of this substance, NADP? We will discuss a little later. As I tell already, pentose phosphate pathway is an alternative route of glucose metabolism because we don't produce energy but produce another very important substances. Now let's tell about these reactions of the first phase. But first of all, let's tell about the role of these very important substances in our body. I tell that NADP H2 has a unique role, but pentose phosphate, the same, has a very important function in our body. You know that pentose phosphate we use for biosynthesis of nucleotides, ATP, GTP, UTP, TTP, which in turn lead to we use for formation of nucleic acid, DNA, and RNA. We use ATP for the formation of coenzyme A, FAD, NAD, NADP, and very important as adenosine methionine. So it's very important role of ribose 5-phosphate. What is the role of NADPH2? First of all, NADP present in our body usually in its reduced form, NADPH, and the ratio NADP plus NADPH is about 0.1. And it means that it present mainly in its reduced form. That's why NADP generally we use for reduced biosynthetic processes. Oppositely of in contract of NAD plus. The ratio NAD plus NADH approximately 1000. You see, it means that NAD present mainly in our body in its oxidative form, NAD plus. And NAD plus mainly play important role in oxidative reaction. We use it for the oxidation. And NADPH we use for synthetic processes. We use it for biosynthesis of fatty acids, cholesterol, nucleotide, neurotransmitters, steroid hormones from cholesterol. But about these processes, we will tell with you a little later, because soon we will have a number of lectures about lipid metabolism. What is additional role of NADPH? We already discussed with you about the role of family of cytochromes P450 of monogenase system. You know that cytochrome P450, microsomal cytochrome P450 with present in endoplasmic reticulum we use for detoxification of different foreign substances, xenobiotics, or end products of our own metabolism with the endogenic substances. If we tell about mitochondrial cytochrome P450, we use it for the start of steroid hormone synthesis, but in both cases, one of coenzyme of cytochrome P450 is NADPH. Moreover, NADPH we use for phagocytosis by white blood cells and oxidant-independent and oxygen-dependent mechanism. NADPH plays the same very important role in biosynthesis of nitric oxide and or with the using of amino acid arginine. And you see that NADPH plays very important role in reduction and detoxification of hydrogen peroxide, H2O2, one of the family of reactive oxygen species, ROS, that are formed from the partial reduction of molecular oxygen. That's why you see that pentose phosphate pathway is very active in tissues and organs where very active, we produce some of lipids, steroid hormones, or fatty acid and cholesterol, and so on. And it plays a very important role in red blood cells. A little later, I explain you why. But first of all, let's tell about the reactions of the first phase of pentose phosphate pathway, oxidative phase. You see, it's rather short way, consists from 3 irreversible reactions and 1 reversible. And during this pathway, we produce 2 molecules of NADPH2 and 1 molecule of carbon dioxide. Now let's tell about each of these reactions, especially with enzymes, coenzymes, and of course with the structure, how you have to study them. First reaction, really first reaction, of course, phosphorylation of glucose. But we start from already glucose 6-phosphate. What happened during the first reaction? During the first reaction happened the oxidation of glucose 6-phosphate. We take 2 this hydrogen and produce 6-phosphogluconolactone. Pay attention on this first carbon. Yes, we oxidize our first carbon of our glucose 6-phosphate and produce 6-phosphogluconolactone. Together with this, we reduce the first molecule of NADP and produce first molecule of NADPH plus H+. Glucose 6-phosphate dehydrogenase enzyme which catalyze this reaction. Pay attention. This and only this enzyme is regulatory enzyme in pentose phosphate pathway. And activity of all this way depend from the activity of this enzyme. Now let's tell, of course, about its allosteric regulation. Mainly activity of this enzyme depends from one of the end product, NADPH. Excess of NADPH lead to inactivation of this enzyme and stop of pentose phosphate pathway, stop of this NADPH production. Oppositely, high concentration of NADP plus lead to activation of this enzyme. It's like feedback inhibition. Both and this NADPH, if we have excess of them, they inhibit first step of pentose phosphate pathway. And high amount of long fatty acid, long chain fatty acid, they're the same, they are inhibitor of this way. So it is inhibited by mainly palmitoyl, the populous fatty acid in our body. That's all. You see, the allosteric regulation of this way is rather simple. Second reaction. During the second reaction, under the action of special enzyme with absolute specificity, lactonase, happened the formation of 6-phosphogluconate, gluconic acid with using of the water. After that, you see that after this hydrolysis, our product already can be present in cyclic form. It leads to ring opening and happen formation of acid. You see, gluconic acid. 6-phosphogluconate we form. This reaction is very simple. Next reaction, but I again repeat this. This and this step, they are irreversible, really irreversible. That's why to write it's irreversible. During the next step happen step by step 2 reactions. First of all, decarboxylation. We remove this carbon dioxide. and from 6 carbon substrate we produce 5 carbon product, ribulose 5-phosphate, ketose, and happen the oxidation again of our 6-phosphogluconate with parallelly a reduction of NADPH2. So during this third reaction, we produce second molecule of NADPH. 6-phosphogluconate dehydrogenase enzyme which catalyzes both steps of this reaction. And we produce pentose, ketose, ribulose 5-phosphate. After that, already reversibly, ribulose 5-phosphate can be converted into ribose 5-phosphate with phosphopentose isomerase. In some tissues, the pentose phosphate pathway ends on this point if we produce enough amount of each of these products, and ribose 5-phosphate and NADPH2. But in many cells that carry out reductive biosynthetic reactions have a greater need for NADPH than for ribose 5-phosphate. And it leads to accumulation of ribose 5-phosphate, a pair of excess of this molecule, and necessary to do something with them. For this, we have second non-oxidative phase. During second phase, we combine together, you see, these pentoses and ribose and ribulose and cellulose, and after some reactions of transferation, later I explain you how, we produce finally or 3 carbon or 6 carbon sugars, glyceraldehyde 3-phosphate and fructose 6-phosphate. You know that they are intermediate of glycolysis and they continue this pathway in glycolysis. All of these reactions, they are reversible. And 2 groups of enzymes we use in this second phase. One of them, they are transketolase. Transketolase carry 2 carbon units from ketose to aldose. And we produce such way triose and 3 carbons product and 7 carbon products like sedaheptulose. And we can use one more variant of enzyme, transaldolase. Transaldolase transfer 3 carbon fragments. For us interesting here, especially interesting, transketolase molecule. Why? Because coenzyme of transketolase is vitamin B1. It's one more function of this vitamin, you see, thymine pyrophosphate. Thymine pyrophosphate has 2 main functions in our body. One of them we discussed already earlier. It's oxidative decarboxylation of keto acid like pyruvate, when we transform pyruvate into acetyl coenzyme A, or the same oxidative decarboxylation but already alpha ketoglutarate in TCA cycle is the same keto acid. And this vitamin B1 is be used in this reaction because it's a coenzyme of the first enzyme in this multi-enzymes complex we discussed 2 lectures ago. And the second very important role of this vitamin in our body, this coenzyme of transketolase is necessary for the second phase in pentose phosphate pathway. Now, so if we necessary a lot amount of NADPH2 and a pair excess of pentoses, after some reactions we can transform, as I tell already them, into intermediate of glycolysis and can use them for energy production. But in contrast, when necessary a lot of riboses for nucleotides or nucleic acid synthesis and necessary greater than NADPH2, we can use this phase, non-oxidative phase, for the production of riboses from fructose 6-phosphate and glyceraldehyde 3-phosphate, only oppositely, because all of this reaction, they are reversible, even without oxidative step. Now, what happened during this second phase? Not me to study this reaction, I only want to show you what happened. Enough if you know only mechanism of second non-oxidative phase and the role of this phase in our body. This second non-oxidative phase, I again repeat it, has two main functions, two main roles. First of all, utilization of excess of riboses, if we need mainly NADPH2, or oppositely, production of these riboses, if in some cells, first of all, we need ribose for biosynthesis of nucleic acid, for example. It's the two functions of this pathway, second phase of pentose phosphate pathway. But what happened here? You see, if we have two pentoses, ketose and aldose. Due to transketolase, it's a transferation reaction, we transfer two carbon fragments from ketopentoses to aldopentoses with biosynthesis, you see, of 3 carbon fragments, glyceraldehyde 3-phosphate, and 7 carbon product. After the 7 carbon, we can interact with next 2 pentoses, you see 7 plus 5. If we combine it with pentoses, together 7 plus 5, we have 12 carbons, and we can divide them into 2 hexoses. Such way, when we combine them together, finally appear trioses and hexoses. Why name of this pentose phosphate pathway is shunt? Really, you see, we have some common point between this way and glycolysis. First of all, the first substrate, glucose 6-phosphate, is the common for both of this way. And for glycolysis and for PPP, pentose phosphate pathway. After that, during the second phase, we produce glyceraldehyde 3-phosphate and fructose 6-phosphate. We after the same continue their way in glycolysis, and we use them for energy production. Moreover, these two ways they depend one from another. If concentration of NADPH in the body is very low, it leads to activation of pentose phosphate pathway. And glucose 6-phosphate in this case mainly we use for, in this way, for the production of NADPH. If oppositely, concentration of NADPH into the cell is high, you know this NADPH is inhibitor of glucose 6-phosphate dehydrogenase. It leads to block of pentose phosphate pathway, and in this case, glucose 6-phosphate already mainly will be used for glycolysis, for energy production. I again repeat this table. It's the main function of NADPH because About this you will listen a little later during next lecture. In lipid metabolism, this part we already discussed. Now let's tell a little about this very important function of NADPH2. We use it for the reduction of glutathione. And what is glutathione? Why it's so important for us? And how glutathione binds with NADPH2. Let's discuss it. Glutathione is a tripeptide. Consists from glutamate, cysteine, and glycine. Pay attention. I hope that you remember yet the structure of amino acids. Glutamate, cysteine, and glycine. Only here very important bond, peptide bond. And this peptide, in the formation of this peptide bond, we use not alpha carboxylic group, but gamma carboxylic group of radicals. This is reduced form of glutathione. Reduced form of glutathione. See on this, reduced. You see? CH. During the reduction of any molecules, later you will see that mainly hydrogen peroxide, happen the oxidation of our glutathione, and two molecules of glutathione join together. A little later I will show you. Two molecules of glutathione join together and we form oxidative form of glutathione with disulfide bond between them. So, due to this hydrogen, glutathione can take part in reduction reactions. You see, glutathione is a donor of hydrogen and protons. First of all, we use it for reduction of hydrogen peroxide and transformation of hydrogen peroxide into the water. Hydrogen peroxide, like another highly reactive oxygen intermediates, can lead to chemical damage of DNA, proteins, unsaturated lipids, unsaturated fatty acids, and finally it can lead to cell death. This reactive oxygen species, they are continuously produced into the cell during different reactions, even during the reactions of the respiratory chain. I already tell about this. And for inactivation of them, we have a special enzyme, glutathione peroxidase, which transform hydrogen peroxide into water. But during, and of course, glutathione is a coenzyme of this enzyme. And you see reduced form and oxidative form. This is oxidative form of glutathione. When we form, you see disulfide bond. So in this form, glutathione is inactive, and it leads to inactivation of full enzyme, glutathione peroxidase. That's why necessary to reactivate it for the prevent damage action of another hydrogen peroxide molecules. How we can reactivate it? For this, necessary to break down this bond and add hydrogens to each of these sulfur of our cysteines. And for the reactivation of this glutathione disulfide, we use a special enzyme, glutathione reductase. And NADPH is a coenzyme of this glutathione reductase. Such way, NADPH plays the role as a source of reduce as a source of electrons and protons for the reduction of hydrogen peroxide, but indirectly through activation of this glutathione peroxidase. And it which is very important for many of our organs, but in our tissues and organs, usually we have not only one source of NADPH2, not only pentose phosphate pathway, we have a number of other reactions which lead to production of NADPH2. For example, malic enzyme, a little later we'll tell about this malic enzyme which is present in the mitochondria. But in erythrocytes, where we have no mitochondria, yes, we have only this way of NADPH synthesis, only pentose phosphate pathway. That's why deficiency or low activity of this first enzyme in this pathway, glucose 6-phosphate dehydrogenase, lead to a pair of big medical problems, especially problem with the blood, problem with the red blood cells, because deficiency of this enzyme lead to lysis of these erythrocytes, lead to hemolysis. You see that about 10% of glucose in the erythrocytes we used for pentose phosphate pathway. You see how many NADPH2 is necessary to produce in erythrocytes for the prevent of harmful action of hydrogen peroxide. This is the main result. I already explained it, that this special system we use for preventing of harmful action of hydrogen peroxide. We have a number of different systems which lead to decreasing of negative influences of hydrogen peroxide and another very active form of oxygen like superoxide radicals and so on. But in the erythrocyte, one of the main way, of course, is we use this glutathione peroxidase and glutathione reductase with NADPH2. Moreover, NADPH2 is necessary for the prevent of oxidation of cysteine in different sulfuric groups of cysteine in different proteins, including hemoglobin. And if happens the oxidation of these sulfhydryl groups, it leads to denaturation, finally, denaturation of proteins and formation of insoluble bodies, insoluble masses called Haynes bodies. These bodies attack membrane of erythrocytes, and finally it leads to formation of more rigid properties of this membrane. And erythrocyte already can't deformate so easily and can't pass small capillaries. And of course they are removed from circulation by macrophages in the spleen and liver. So NADPH2 plays many very important role in erythrocytes. You see in this scheme you can see what happened. Concentration of oxygen in erythrocyte of course is very high, and it increases the risk of formation of superoxide anion, which under the action of special number of enzymes can be converted into hydrogen peroxide. But hydrogen peroxide, the same, is a harmful product. We already tell. It leads to formation of Haynes bodies, you see, and it leads to activation of membrane lipid peroxidation. It's a cascade mechanism. And it finally leads to lysis of this membrane, damage of this membrane, and lysis of erythrocyte. That's why necessary to transform this very harmful molecule into water. And we can do it with this, you see, glutathione peroxidase. But during this reaction, detoxification of hydrogen peroxide, happens the inactivation of glutathione. And for reactivation of this enzyme, you see, we use glutathione reductase. And coenzyme of this enzyme is our NADP H2, which we produce due to pentose phosphate pathway. Deficiency of this enzyme lead to block of this finally of glutathione peroxidase, decreasing of its activity, and accumulation of hydrogen peroxide. And one of the main symptoms of this enzymopathia is hemolysis. We discussed with you all main pathway with carbohydrates, and we tell about glycolysis and about metabolism of glycogen and gluconeogenesis and pentose phosphate pathway, and every time we discussed allosteric regulation. I think that you remember that what is allosteric regulation? We have a number of regulatory enzymes in each metabolic pathway. And when any factors, activators or inhibitors binds with a special regulatory site, allosteric site, it leads to change of these enzymes conformation, three-dimensional conformation, and of course change of conformation of active site. And as a result, we change activity of this enzyme. But it's a local regulation of any special way in any special cells. And meeting of cells regulate processes due to this mechanism. Oppositely, we have a general regulation on organism level, and we can regulate main direction of our metabolism. And generally, we regulate the level of glucose in the blood. And of course, we can do it with hormone regulation. What hormones took part in regulation of carbohydrate metabolism? See, almost all of them. And insulin, and glucagon, and thyroid hormones secreted for, and cortisol, because it's from even the group glucocorticoids. Glucocorticoids already show us that these hormones mainly took part in metabolism of carbohydrates. And epinephrine, of course, and growth hormone, and prolactin. All of these hormones mainly took part in regulation of the level of glucose concentration in the blood. Only pay attention that only insulin leads to decreases of glucose level in the blood. All other hormones, they are antagonists of insulin. They increase level of concentration of glucose in the blood, until normal, of course, if we tell about healthy organism. How they do it? Let's discuss. Let's start from insulin. And glucagon. Why we start from there? Because of course they are top. They are major hormones which we use for regulation of carbohydrate metabolism. They play central role in this regulation. That's why we will tell about these hormones detailedly. As we discussed, insulin, the main effect of insulin, it's decrease the level of glucose in the blood, and insulin mainly used most prominent in three tissues: liver, muscles, and adipose tissue. In the liver, muscle, and muscles, insulin increases glycogen synthesis. In the muscle and adipose tissue, insulin increases glucose uptake by increasing of the number of special glucose transport protein, GLUT4, in the cell membrane. In the liver, insulin decreases the production of glucose through the inhibition of glycogenolysis and gluconeogenesis. Glucagon. Glucagon has opposite of insulin action. Glucagon stimulates production of glucose due to glycogenolysis, degradation of glycogen. Insulin increases production of glucose from non-carbohydrates. And you remember the name of this pathway is gluconeogenesis. How these hormones act? Of course, they increase or decrease activity of corresponding enzymes. How? Let's discuss it. First of all, you know that both of them, they are produced in the pancreas. Only insulin is produced in the beta cell, glucagon is produced in the alpha cells. They are really endocrine hormones. They have endocrine mechanism of action. What it means? That after production, they are released into the blood. Through the blood, they achieve target organs where they interact with special receptor and achieve any effect. We will see any cells answer. But they have an paracrine action. It means they regulate production of hormones inside of the pancreas. Such way, insulin decreases production of glucagon, upside down. Glucagon decreases production of insulin due to paracrine action without releasing in the blood. The act of neighbor cells. Insulin. Wait a little later, next semester, we will again tell about hormones and about their role in our body. On that lecture, we will tell about all way of insulin activation. It's 2 steps of limited proteolysis, but finally, insulin consists from 2 chains, you see, we join together with disulfide bonds. Insulin, of course, influences not only on carbohydrate metabolism. Insulin, you see that it, first of all, decreases production of glucagon in alpha cells, and insulin has mainly anabolic effect, and it increases not only carbohydrates, regulate not only carbohydrate metabolism, but increase uptake of fatty acid and amino acid in the tissue, increase lipogenesis, as you tell. But today, now, we tell about carbohydrate metabolism. That's why mainly we will tell about effect of this hormone on carbohydrate metabolism. First of all, insulin increases the uptake of glucose into the cell. It's the fastest effect of insulin during a few seconds. After that, insulin stimulates hormones of glycolysis, dexokinase, phosphofructokinase, and pyruvate kinase. Insulin increases activity of glycogen synthase and the increased production of glycogen. And of course, insulin blocks glycogen phosphorylase and prevent degradation of glycogen. It's a main scheme, you see. Maybe such way it's be easily to remember. The main function of insulin is decreasing of glucose concentration into blood. And of course, insulin will stimulate all processes which lead to decrease of this glucose concentration in the blood. So it's increased uptake of glucose into the cell and immediately stimulation of glucose conversion into the glycogen or pyruvate glycolysis. So insulin will activate both of these processes. And of course you have to remember the main regulatory enzyme of each of these processes. Here this is glycogen synthase, here 3 kinases: hexokinase, pyruvate kinase and phosphofructokinase. And of course insulin will stop production of glucose. How insulin acts? Very interesting. It's a special absolutely mechanism, because insulin receptor, it's a unique machine for insulin action. Insulin receptor consists from 2 symmetric parts. You see this one and this second. Each part consists from 2 subunits: alpha, which present in the cytosolic part, and beta, with the same consists from hydrophobic part, which is incorporated into membrane, transmembrane part, and this special cytosolic part of beta subunits, which has tyrosine kinase activity, this cytosolic domain. Now, in cytosolic domain. Now, see, when we tell about alpha subunits, they have high specificity for insulin. That's why they can recognize and bind insulin. And this binding with insulin lead to activation of these cytosolic subunits, and because these subunits consist tyrosine, and tyrosine has a kinase activity. What is kinase? You know that kinase enzyme that took part in phosphorylation with using of ATP. And after activation of this part of receptor, it leads to, you see, phosphorylation of these subunits and activation of these subunits. This activation of beta subunits lead to phosphorylation of a family of proteins called insulin receptor substrates. Insulin receptor, you see, insulin receptor substrate. A number of proteins with the same will phosphorylate, and they becomes active. These proteins lead to, for example, activation of protein kinase, lead to inactivation of protein kinase, but you see activation of protein phosphatase. Phosphatase enzyme will take part in dephosphorylation. Protein shows us the name of substrate, what we will dephosphorylate. So under the activation of this protein, it leads to activation of protein phosphatase, and protein phosphatase in turn lead to dephosphorylation of glycogen phosphorylase. And such way inactivation of this enzyme and stopping of glycogen degradation, and such way activation of glycogen synthesis, because dephosphorylation of glycogen synthase in turn lead to activation of this enzyme, and such way stimulation of glycogen synthesis. After that, it's one of the effects. After that, you see that phosphorylation of these beta subunits, and after the phosphorylation of these proteins lead to activation of translocation of GLUT proteins, which are present inside of cells in a special vesicles, and they are transported to the membrane, and these GLUT proteins, they are incorporated into membrane, of the cell, and such way we activate transportation of glucose into the cell. Is the fastest effect. But we don't not only activate and change activity of different enzymes. Some of them activate, some of them inactivate. Insulin has one more effect. It regulates gene transcription, and such way it's to part in regulation of biosynthesis of proteins, and finally amount of different enzymes. So different, that's the slowest effect, which appear after some times. You see, it's so many different levels of insulin action. Membrane effect, changing of enzymes activity, and changing of enzymes concentration due to effect of insulin on gene transcription. Due to this, insulin finally decrease the level of glucose in the blood, because we can't simply to carry this glucose from blood into the cell. If we simply increase concentration of glucose into the cell, it leads to increase of pressure inside of the cell, and finally can lead to damage or analysis of the cell. That's why immediately necessary to do something with this glucose, and concentration of glucose into the cell, the same must be constant. In such way we can use this glucose or for glycogen synthesis or for energy production. First of all, it depends, of course, from the needing of the cell in this moment. If concentration of glycogen is enough already for the cell, if we tell about muscles or liver, excess of glucose already after that we will use for biosynthesis of fat through acetyl coenzyme A formation. Now glucagon. Glucagon the same as a protein, and glucagon has opposite effect. It's an antagonism of insulin. First of all, high concentration of glucagon lead to block of insulin synthesis and activation. How we activate glucagon synthesis? What is stimulator of glucagon synthesis? Low level of glucose in the blood, less than 3.3. It leads to activation of glucagon synthesis, and glucagon concentration of glucagon is increased, and glucagon already starts its function. It's activate all enzymes and all metabolic ways which lead to glucose production from carbohydrates, glycogen, or from non-carbohydrates, gluconeogenesis. And of course, glucagon will stop all processes of glucose utilization. So glucagon decreases activity of glycolysis and glycogen synthesis. But mechanism of glucagon action is a classic mechanism of endocrine hormones action, peptide hormones action. It's a mechanism of second messenger, mainly catabolic hormone. You see that glucagon increases the blood level of glucose by stimulation of glycogen breakdown in the liver, and by stimulation of gluconeogenesis, the same in the liver, and by inhibition of glycolysis and glycogen synthesis. The same scheme. Glucagon upside down, increase concentration of glucose. So it will be activate all processes for glucose synthesis. This you see, in glycogenolysis and gluconeogenesis, and decrease using of glucose in any direction. What is the mechanism of glucagon action? As I tell already, it's a classic mechanism of hormone action. When glucagon interacts with its receptor, it leads to activation of When glucagon binds with its receptor, special receptor, it leads to activation of G protein. When both three subunits of G protein they are together, it's inactive form. After activation, when glucagon binds with its receptor, alpha subunit is separated, and it's already active form. For this separation, you see, we use GTP. See here. That's why full name of this protein is G protein, GTP. First letter here. These active alpha subunits, after that, interact with the special adenylate cyclase, the same membrane binding enzyme, and activate this adenylate cyclase. The main function of adenylate cyclase is formation of cyclic AMP from ATP. Cyclic AMP, for cyclic AMP, interact with protein kinase A and activate it. You see, it's inactive protein kinase. This active when cyclic AMP binds with it. Active protein kinase lead to phosphorylation, initiate cascade of phosphorylation of other proteins. First of all, it leads to the phosphorylation of glycogen phosphorylase kinase phosphorylation. And from glycogen phosphorylase kinase B, it is converted into glycogen phosphorylase kinase A, active form. This glycogen phosphorylase kinase leads to phosphorylation of glycogen phosphorylase, and glycogen phosphorylase B, inactive form, is converted to glycogen phosphorylase A, active form. And after that, this active glycogen phosphorylase starts degradation of glycogen with the formation of glucose 1-phosphate, which after that is converted to glucose 6-phosphate, and finally simply glucose in the liver. And this glucose go out from hepatocyte into the blood, in such way we increase the level of glucose in the blood. It's a classic mechanism of second messenger. Epinephrine has absolutely the same mechanism. Epinephrine, the same bind with this receptor and activate this mechanism. Only glucagon and epinephrine has some defects because they have different target organs. But about this a little later. What else hormones took part in the regulation of it's one more very good scheme, and I want to show you again that mechanism the same, but due to phosphorylation of different enzymes, you see, we activate glycogen synthesis and inactivate, you see, phosphorylation happens, But when we phosphorylate glycogen phosphorylase, it becomes active. When we phosphorylate glycogen synthesis, it becomes inactive. That's why glucagon, like epinephrine, they activate degradation of glycogen and block glycogen synthesis. When the level of glucose becomes very high after eating, for example, we activate insulin. Insulin, after cascade of mechanism and final activation of protein phosphorylase, lead to dephosphorylation of this enzyme, and after that we stop degradation of glycogen and oppositely activate this process. You see, very simple processes. Phosphorylation and dephosphorylation of enzyme. It's a covalent modification of enzyme, and due to this we change their activity and change direction of our metabolism. So the same here, absolutely the same. You see, we activate formation of glucose, and after the glucose go out from hepatocyte into the blood and inactivate glycolysis. Due to phosphorylation of pyruvate kinase. It's a function of glucagon. Epinephrine has mechanism of action like glucagon. Growth hormone, the same is antagonism of insulin, and the same took part in regulation of gluconeogenesis and glycogen metabolism. Glucocorticoids, which is secreted by adrenal cortex. This hormone increase gluconeogenesis, and especially gluconeogenesis. If glucagon mainly increase gluconeogenesis from lactate, pyruvate, glucocorticoids, cortisol, mainly increase production of glucose from amino acids, gluconeogenesis from amino acids. Here we tell about thyroid hormones. They are T3 they are the same antagonism of insulin. So the abnormal and high concentration of this hormone can lead to increase the level of glucose in the blood. You know that epinephrine hormone of acute stress, and usually if we check blood after strong acute stress, we will see increasing of the glucose level in the blood. But after some time, of course, it becomes normal. Cortisol, it's hormones of the chronic stress, and high concentration of this hormone in the blood, Cushing syndrome, lead to continuously high level of glucose in the blood. And sometimes we even use a special name, steroid diabetes, and high level of glucose, but not due to low insulin, but due to high amount of cortisol in this organism. Epinephrine and glucagon. What is the difference between them? They have, you see, mainly they have different target organs. Epinephrine stimulates metabolism in the muscles. And you know that there is no any gluconeogenesis in the muscle. That's why epinephrine mainly act on glycogen metabolism. It's increased glycogenesis. It's increased, yes, glycogenolysis, degradation of glycogen, with the stopping of glycolysis and glycogen synthesis. Glucagon mainly acts on the liver. That's why glucagon will stop glycogen synthesis, glycolysis, the same, activate glycogenolysis in the liver, degradation of glycogen, but together with this, glucagon activate gluconeogenesis. Because you know that gluconeogenesis occurs mainly in two organs. First of all, of course, in the liver, about 90% of all gluconeogenesis, and in the kidney the same, but the role of kidney in gluconeogenesis is not so important. Normally, only about 10% of glucose is produced in the kidney due to gluconeogenesis, and usually for the kneading of kidney itself. Also, first I give you this lecture. It's rather short, but we discuss with you all material about carbohydrate metabolism. I hope that you understand everything. If you have any question, you can ask me now. We have a time to answer, because from the next week we start a new cycle of metabolism. during 5 lectures, we will discuss with you the specificity of lipid metabolism in our body. Now we have a few time for questions. You can ask me if necessary. If you don't understand anything, I have a time to answer. If no, that's all for today. And I want to tell you goodbye. Try to study better. Try to study all this metabolism, all this way. Necessary to know the structures, and if you will prepare this metabolic way for our control point and exam, of course necessary to know all metabolic way with the structures, with the name of enzymes, with the name of regulatory enzymes, and of course the regulation and allosteric regulation and hormone regulation. And as usually, because we are with you, we are doctors, though necessary to know normal condition of this metabolism and disorders of them with little different diseases. That's all. I don't see any questions. You don't ask me. I hope that everything is understandable for you. That's all for today. Goodbye, dear students.